cloud. Okay, so one of my favorite chapters, okay, to talk about DNA technology. Okay, all right. DNA technology is changing the world. That's why you guys all heard of DNA. That's why you guys probably heard at least a little bit about everything we're talking about. Maybe you guys don't know the big details and stuff, like small details and stuff, but the big details you probably know already. But we'll get into many, many, many things. Okay, so DNA is used as a powerful tool in research, in medicine, agriculture, criminal justice, just about anything you can think about, actually. Okay. So scientists can manipulate DNA. We can change the sequence, we can copy it, turn it on, off, etc. Okay. So that's it. Okay. So we can change DNA. If we change DNA, then we can change the protein, and then we can manipulate entire organisms, and we can look at how things are done. Okay. So the first thing I want to make sure you guys understand is what is a transgenic organism. Okay, a transgenic organism, by definition, is a organism that receives a piece of recombinant DNA. So a gene or genes from other sources. Okay, so you can look at this fish. This fish, okay, is a transgenic organism. Okay, it's designed as a pet shop fish. Okay, call it blowfish. Okay, so what they did was they took DNA from a jellyfish and inserted it into a regular colored fish, and therefore now that fish glows in the dark, and you can make it glow different colors. Okay, so you can make it glow green, blue, pink, purple, etc., etc., etc. And every year they come up with new organisms. Okay, so you can see the normal. This is their normal color, and then they can glow different colors. And now they have different types of blowfish, and stuff like that. So, I mean, that's not really that interesting. It's important. It's fun. And it's for pet shops and stuff. But let's talk about real transgenics and how we can modify the world using transgenic organisms. Okay. So bacteria are the most common transgenic organisms because bacterial DNA is easier to manipulate than other DNAs. Okay. So insulin, blood clotting factors, fertility hormones, okay, that are used in as birth control pills and other stuff. They're all produced in bacteria. Okay, so you take the genes that are responsible for making these hormones and you put them in bacteria and you just grow them up in large fats. Okay, then these bacteria will naturally produce large amounts of these things, and then you can extract them. Okay, other ways you can use transgenic bacteria. You can engineer them with genes to degrade stuff like petroleum, okay, and pesticides and other soil pollutants, and all you gotta do is dump the bacteria onto the soil or onto the oil spill, and the bacteria will slowly eat away at oil, okay, until there's no more oil. So scooping oil out of water is freaking impossible, right? But if you dump bacteria on the surface of it, and let the bacteria chew up the oil. That makes cleaning up a lot easier. Okay. Okay, so how we do this is we take plasmids, okay, there's a small circle of double-stranded DNA that's separate from the chromosomes. We add whatever genes that we want to the plasmid, okay, and that's called a recombinant plasmid, okay, and we use restriction enzymes to cut it, okay, and then we insert it into the host. One of many different ways, okay. The zapping the cells with electricity is the easiest way, but there are other things. Okay, we can insert that into plants as well, so we can alter tobacco plants and stuff. Okay, so this tobacco plant, for example, is herbicide resistant. So you can grow plants in herbicide that will kill other plants, but not the tobacco plant. So the tobacco plant will grow. Okay. Okay. So what's even cooler is you can grow in tobacco plants like this. Okay, like this guy right here. And eventually, if we do this right, and this is hopefully what will happen in the future, if we do this right, we can actually make lamps out of full green plants. And instead of using them, instead of using streetlights, we use plants that glow in the dark. Okay. We'll skip this part about viruses. Okay. All right. So, okay, make sure you answer these two questions. Okay. The next thing we can do with DNA is understand the sequence. Okay. We can genetically sequence your DNA. Okay. Okay. So DNA sequencing reveals the order of the bases. Okay. DNA technology is not a lot cheaper. So the cost of sequencing a human genome plunged from about 100 million in 2001, which is the original genome project. Human Genome Project about $1,000 in 2017. The sequencing is just a pretty easy standard thing to do. Okay. Okay. The next thing that definitely need to understand is what PCR is. Okay. Because PCR is by far the most important concept in DNA in the last 30 years. Okay. If you know the sequence of DNA, you perform a technique called polymerase chain reaction. But PCR rapidly produces million copies of the selected DNA sequence in a test tube, and then give investigators enough DNA to analyze. Okay, so if you have a small piece of DNA and you want more of it, you can use PCR to increase it. Okay, so you can make millions of copies using a very special DNA polymerase. Okay, and copy. So you put it through this process. So one round, okay, you heat it up, you lower the temperature, you amplify the sequence, you get 2 copies. In 2 rounds, you get 4 copies. 3 rounds, you get 8 copies. 25 rounds, you get a million copies. Okay, 40 rounds, you get a billion copies. So it is a lot of DNA. You get a lot of DNA very quickly. Okay. Okay, so investigators use PCR to amplify DNA from tiny biological samples, such as those left at a crime scene. Okay, I use PCR to amplify DNA from whatever fly I wanted to look at. Same thing, small piece of DNA is enough. Okay. So DNA profiling examines short tandem repeats between people. So everyone has these short tandem repeats. Okay, they're just small little sequences that repeat over and over and over again in the human genome, but everybody has different number of repeats. So some people have 7 repeats, some people have 9, some people have 8, some people have 6, some people have 10, some people have 20. So different people have different amounts of short tandem repeats in different locations. And so I can look at a couple of those locations and see what the genetic profile is. And so here I can match the DNA profiles of a whole bunch of different locations, okay? And then these are the repeats. And you can see for this particular one, this, okay, only matches this number 2. So number 2 was at the crime scene. And you can see the DNA from everywhere on this thing matches everywhere on this thing, but it matches some of the other stuff, but this is an exact match. So if this is an exact match, then you know this is a person who was actually there. When you're doing this for real, they tell you something like, "Oh, this is 1 out of a billion people who can match this profile," because 1 out of a billion has this combination of DNA. So if it's 1 out of 10, then yeah, it could possibly be someone else, right? Because 1 out of 10 is not impossible, but 1 out of a billion is really rare. Okay. So you can use DNA profiling to strengthen the DNA criminal justice system. You can not only find the guilty person, which is important, but you can actually exonerate people who are, who have been found guilty. Okay, like this guy right here. Okay. Okay. Make sure you can answer this question as well. Okay. Stem cells and cloning. Okay. So stem cell research often sparks public debate. Some see it as useful technology, some see it as unethical. Okay. So let's talk about what stem cells actually do. Okay. So animal development relies on stem cells. Okay. Stem cells can differentiate into many, many, many different types of cells. Okay. And this is especially true of embryonic stem cells. Embryonic stem cells are totipotent, which means they can differentiate into all the different types of cells in the body. So if I take embryonic stem cells, I can develop them into brain cells, I can develop them into heart cells, I can develop them into kidney cells, I can develop them into muscle, I can develop them into bone, all relatively easily. Okay. Adult stem cells are pluripotent. Pluripotent only allows you to differentiate into some cells, like bone marrow can differentiate into red blood cells and white blood cells, but not very much other stuff. Okay. Okay, cloning is okay. Or asexual reproduction is common among single-cell organisms. Plants are very easy to clone in the laboratory. Using a couple of cells, you can grow them very easily. But animals are different. Okay. Somatic nuclear cell transfer, okay, is used to clone animals. So when I was in college, that was when they first cloned Dolly. That was front page news in any science journal ever. Okay, the nucleus from a sheep cell was used to create Dolly. The DNA was identical to the donor. Okay, so you can clone sheep. You can clone sheep. You can clone humans. Humans are not any different than sheep when it comes to like simple genetics. Okay. Okay. So new animals can be cloned from adult cells. Okay. We've cloned basically a lot of different animals already. There are some places where it is useful to clone animals. Okay, like for example, if you wanted to clone your favorite cow, okay, that if you have a cow that produces a lot of milk or produces good beef, you might want to clone that cow. Okay, instead of breeding it, because breeding it only gives half enough DNA to the offspring. Cloning it gives all DNA to the offspring. Okay, so a lot of there's a lot of companies that invest in that because you find your cow that's giving great milk, then you want to make a copy of it, okay, instead of breeding it. Okay. Okay. The second place, pets. Like people that attach to a particular pet and they if one dies, they might want to clone it and make a copy of it. Okay. Please understand a pet's personality is not cloned in any way. Okay. Pets will have their own independent personality, but they'll look similar and have very similar characteristics to adult physical characteristics. Okay. And some people really want that. Okay. And they're worth paying, they're willing to pay. The last time I checked, it was about 20K to clone a pet. Okay. But hey, you have 20K to waste, go ahead. Okay. Okay. Is human cloning ethical? And we will discuss this in class. Okay. All right. Okay. Okay. So DNA probes can be used to detect different sequences. Okay. So this type of probing is called FISH, fluorescent um, fluorescent hybrid oh, fluorescent in situ hybridization FISH. Okay, so I take a label that's fluorescent and I bind it. So this sequence will only attach to the sequence that I want. So I'm looking for the sequence of a person who has cystic fibrosis. So if this glow lights up, then this person will have cystic fibrosis. So I'm looking at this person right now, and I can tell this person will have cystic fibrosis right now or in the future. And this where the technology is used is like in utero. Okay. So I could identify cystic fibrosis, hemophilia, Tay-Sachs, sickle cell. Okay. I can also do pre-implantation genetic diagnosis. We can look at embryos that are very early. So after fertilization, these like the eight cell stage occurs in the petri dish. So you can take one of these cells away from the other seven, and you can look at it. And remember, all 8 cells will have the same DNA. So you can actually analyze the symptoms and see what if they have any. Okay. Okay. And then gene therapy. Okay. Gene therapy is the latest and greatest. We're quickly developing new technology. Okay. The overall goal is to replace faulty genes with good ones. Okay. Okay. Especially using CRISPR-Cas9. Okay. And ethical issues. Okay. We're going to talk about this in class. Okay. So, okay. Cool. Okay. Hopefully this is a short chapter, so hopefully you guys understand it. Anyways, I will see you in class.